models. And what I'd like to talk to you about today is what can we learn from the previous pandemic and how we can take those lessons to prepare for future pandemics. So, um, so to start with, we need to understand what defines a pandemic. And, you know, the key point is that during a pandemic, a disease spreads out quite uncontrollably across countries and continents, and it does significantly impact all aspects of society, from socioeconomic to the health of individuals. And, pandemics have happened before in history, quite famous examples. We have the 1918 Spanish flu, but also recent pandemics such as the SARS or MERS pandemics, which indeed were more localized and didn't spread out over the entire globe. However, they were caused by viruses very similar to the SARS-CoV-2 virus. So this goes to tell us that it is important, when a pandemic happens, to draw certain conclusions and prepare our efforts for the next pandemic in order to try to prevent it. And one of the key reasons why COVID-19 indeed became a pandemic was its high transmission rate, and we do live in an age where every country is connected to one another. And to make matters worse, many individuals that were indeed infected with the SARS-CoV-2 virus, they were asymptomatic spreaders. So in order to look at the key scientific lessons from COVID-19, we need to understand the virus itself. And the virus, or the SARS-CoV-2 virus, was able to do an excellent job at infecting humans thanks to its spike protein, which enabled virus entering through human cells. And the mode of transmission varied from respiratory droplets and aerosols to direct contact between an infected individual and a healthy one. And of course, there have been super spreading events such as single individuals that were infected, many others in crowded areas such as aeroplanes or buses or even classrooms. And as the pandemic went on, of course, mutated variants of the virus emerged, and while these were less deadly, there were variants with higher transmissibility, such as the Delta or the Omicron variants. And the key point here is that it is relevant as a pandemic progresses to monitor it properly and to understand the key changes that can occur. And before we delve into the more scientific response to the pandemic, we also have to look at it from a mathematical point of view. And here, the role of data in modeling plays a crucial part. And just to break down the basics of epidemiological modeling, we have this concept of R0, which is the basic reproduction number, and that measures how many people are infected by one diseased individual. And the goal is to reduce R0 below 1 to control the spread. If R0 is above 1, then we can end up in a situation where we have an exponential increase of the cases. And once we have this understanding of modeling, the next step is, of course, to collect data and analyze it. And the key metrics here would be case numbers, hospitalization numbers, and unfortunately, of course, deaths as well. And the purpose would be to identify the key trends and predict what the local needs are for the healthcare system. And at the same time, predictive modeling is an extremely useful tool because it can forecast scenarios based on the data available, and that would also support resource allocation and policy decisions. And here, I do have a question for all of you. I will add it in the chat. Essentially, like, try to imagine that you're part of a team using data to predict the course of a pandemic. What data do you think is the most important data to track, and why? Now, as promised, moving on to the scientific response to the COVID-19 pandemic, there have been two main approaches to try to control it and stop it. The first one, vaccines, which indeed have been a landmark development in the COVID-19 response. And quite astonishing. It was a rather astonishing feat, the development of the first mRNA vaccine. Typically, a vaccine would take 5 to 10 years to be developed in order to check out all the safety requirements. However, due to the stress imposed on society, a lot of resources have been allocated to the development of vaccines, and the first vaccine was developed in just under 12 months. And one famous example would be the Pfizer vaccine, which is an mRNA vaccine. And the way it works is that scientists create this synthetic mRNA strand, which essentially instructs the cell to produce a certain part of the virus. What we would be interested in would be the spike protein. As a response to that, the immune system creates antibodies that are able to identify the spike protein of the SARS-CoV-2 virus. And once the virus enters and infects an individual, the antibodies are able to identify the virus and neutralize it. And while vaccines have been indeed very efficient, the reality is that the more people got infected, the more mutants evolved. And in that scenario, early generation vaccines became redundant. At the same time, vaccines have not been available everywhere across the world, or individuals could not take them as a preventative measure, either due to personal beliefs or due to pre-existing medical conditions. So in a scenario like that, of course, we want a multi-pronged approach, and that would be represented by drugs, which would be used to treat individuals once they have been infected. And in terms of developed drugs, we again have notable examples such as small molecule inhibitors, such as nirmatrelvir, which is a peptidomimetic molecule. So just to break down the entire drug design of this revolutionary medicine, what you see here on the bottom left corner is the crystal structure of an enzyme called the main protease. And the main protease plays a crucial role in the viral replication of the SARS-CoV-2 virus. However, if you are able to neutralize the activity of this enzyme, then the virus cannot replicate anymore, and then it dies out. And in order to fulfill its activity, the virus has to accept at its active site its natural substrate. However, if you block this active site with an inhibitor, which is resemblant of the natural substrate, then we can stop the activity of this enzyme. And that is precisely what this drug developed by Pfizer achieves. For those of you that are familiar with functional groups in organic chemistry. This group here, the nitro group, is the reactive warhead that irreversibly binds to the in the active site to one of the amino acid residues, and thus inactivates the enzyme, and no virus replication would occur afterwards. Another approach to treating the individuals with COVID-19 infections would be represented by monoclonal antibody treatments. And here, the idea would be to administer to individuals synthetic antibodies that target the virus directly. And typically, you could target various parts of the virus. However, as mentioned beforehand, the spike protein that plays a key role in entering for the virus to enter the human cells. So when the synthetic antibodies bind to this to the spike proteins, then the virus becomes bound to the antibodies. And once that occurs, of course, the T cells recognize these complexes, then they assimilate the virus and destroy it. And here we have multiple examples, such as drugs developed by Eli Lilly or Regeneron. And indeed, during the early days of the pandemic, this approach was effective because it has been used before, and ideally you do want a medicine that has an established safety profile, but at the same time, monoclonal antibody treatments also had their limitations. For example, when being developed, you only have available the current strains that are circulating among the human populations. However, as I mentioned before, once the virus starts to mutate, then these antibodies, which were developed for previous variants, would of course have reduced efficacy against these highly mutated variants. And indeed, when the virus mutates, it tends to mutate in key areas where it suffers evolutionary stress. For example, if an individual has this sort of immunity against the SARS-CoV-2 virus by developing antibodies or some sort of immune response with respect to the spike protein, then the virus would modify its structure accordingly at the site of the spike protein. However, in the previous example, when we're looking at antiviral drugs that target enzymes such as the main protease, of course, the way the viruses adapt to this evolutionary stress is by modifying their active sites, and sometimes certain key amino acid residues can become mutated, and in that scenario, your drug doesn't work anymore. And just to further understand the why it doesn't work anymore, just try to imagine of a lock and key, where the lock is your enzyme and the key is your drug. And of course, there can be only one key that unlocks that lock. Now, and typically that key would be your natural substrate. However, you can mimic that key by using this drug, such as nirmatrelvir, and you can block off the active site in this sort of tight model where, like the lock and key model. But then, as I mentioned, if you mutate the active site, and you preserve a certain sequence that works for the natural substrate, but not for the drug, then resistance emerges for latest generation drugs such as mirmatrolvir. And that is indeed a huge challenge in terms of going forward and trying to develop medicine for infected individuals with a certain virus. Um, early early during the pandemic, like it needs to be emphasized that there weren't that many drugs available. And in order to develop new drugs, of course, a lot of time and effort and resources are required. And in order to achieve the like the desired outcomes, of course, this new medicine needs to be tested in clinical trials. You need to find volunteers, and of course, you need to establish like a safety profile that is both efficacious and 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 cost effective. Certain drugs, such as antibody-based drugs, while they can be effective, they can also be very costly to make. But the main restriction in terms of developing a drug for COVID-19, especially in the early days of the pandemic, was time. There was an urgent need for new medicine. And in order to alleviate the situation and offer some relief, especially for individuals that perhaps ended up in A&E with severe respiratory problems, a different sort of approach was taken, and that was to repurpose drugs as COVID-19 treatments. And what is drug repurposing? Essentially identifying new uses for existing drugs to treat diseases outside their original purpose. And while this accelerates treatment availability thanks to established safety profiles, the results might not always be the ones that one would expect. And the reason for that is because these drugs, because they were developed for other diseases, they might not work as well for COVID-19. And if you recall my lock and key analogy with the main protease, the key enzyme responsible for the viral replication of the virus, you will recall that specific molecular structures would be required to fit in the active site of an enzyme. So these drugs, such as dexamethasone, originally used for inflammatory conditions, and indeed, this drug was found to reduce mortality in severe COVID-19 cases. While being efficacious in that sense, it had its own limitations because indeed it wasn't designed to tackle the specific enzymes that are present in the SARS-CoV-2 virus. Another example of drug that has been used as a repurposed drug would be remdesivir. Again, initially developed for Ebola, another virus, and this was shown to reduce recovery time in hospitalized patients. So, in a nutshell, repurposing drugs, it is a very cost-effective measure. They are quick to deploy in clinics, but they do have variable efficacy against COVID-19. And here I do have a question for everyone, so feel free to just jump in the chat. Do you think there are any possible problems with repurposing drugs besides the mentioned ones? I'll just give you 2 or 3 minutes, and then we can go through the answers and also elaborate a bit more on my own um, ideas here. Perhaps any legal issues or ethical issues. Just checking the previous answers. Yes, that's absolutely correct. In terms of the data that you'd like to collect, transmission rate, very important, mortality rate. Uh, Indeed, number of people who have immune disorders, immune system disorders, and as well the location of the latest cases. Yes, you do want to um, build a map of where these cases occur, but also how they are connected to one another. And then among those populations to break down the various factors that contribute to um, how this how the spread can occur. Yes, that is indeed a very valid point on my latest question regarding the issues in the repurposing drugs for COVID-19. Intellectual property and patents, they can pose a significant problem to repurposing drugs. Um, and the reason for that is twofold. Firstly, a big pharmaceutical company might hold the rights to a specific drug. And in that scenario, they can control who has access to this drug, who can manufacture it, and even more importantly, who can modify it in order to make better variants of this drug. Essentially, a patent covers not only the molecule of interest, which would be, let's say, dexamethasone and its various structure motifs, but also variations on its core. For example, instead of having a fluorine atom as substituent, we can have a chlorine atom. And instead of having a ketone over here, we can have an oxime, and so on and so forth. And that is indeed really important because we do want to be able to optimize our drugs for our very specific needs. However, we might not do without prior agreements with the companies that own the intellectual property rights for these derivatives. Any other issues that you can think of regarding repurposing drugs? Perhaps on the ethical side of things? I think that um, a key factor here, ethically speaking, is drugs, before they are being approved for a specific indication, they have to go through an extensive clinical trial where, like, you do have a double-blind sort of trial where you give patients the drug and also the placebo. And in that scenario, you can see how effective it is. And without having that data, sometimes you can end up in a situation where you can take rash decisions in terms of what would be the best treatment. Let's just imagine a situation where a doctor has both dexamethasone and remdesivir as potential treatments for a patient. Which of the two should the doctor choose? Should they choose the dexamethasone or remdesivir? And here, of course, it is important to look at various factors such as patient medical history or their age or how they typically progress through their COVID-19 treatment. However, without this sort of extensive clinical trial data, it can be a bit of a gamble deciding what is the best course of action. Moving on to scientific innovations that occurred during the COVID-19. So besides the obvious a revolutionary medicine that has been developed, such as vaccines, drugs, and so on and so forth. Another revolutionary part about this global health crisis is that it was also a catalyst for scientific innovation. And the pandemic indeed accelerated advancements in technology, diagnostics, and research at an unprecedented scale. And looking at some of these innovations, we can point out innovations such as rapid genome sequencing and variant tracking. And here, within days of identifying the COVID-19 virus, scientists were able to not only sequence the virus's genome, but also share it globally. And making that sort of information readily available for the scientific community was essentially a crucial step for vaccine development, for diagnostic and understanding how the virus spread. And furthermore, being able to track in real time variants such as the Alpha, Delta, or the Omicron variants, that was only possible through global genome sequencing efforts. And perhaps it's not quite clear what revolutionary step this was, mainly because everyone had a rapid test available or even undertook a PCR test when needed. But just to put everything in perspective, this sort of technology 20 years ago was prohibitively expensive and not easy to manufacture or deploy. Whereas nowadays we are able to produce a lateral flow device and distribute it everywhere. We can find it in pharmacies at a very affordable price. And this indeed has played a key role in helping to manage the pandemic. And in terms of these advances in diagnostic testing, PCR test became the gold standard for detecting the virus because using a PCR test, you can identify small amounts of viral genetic material with high accuracy, whereas the rapid antigen test, which provide quick results within minutes, and these are indeed convenient for large population, they're not quite as accurate. So that would be a drawback of using rapid tests. But of course, the drawback of using PCR tests is it takes a longer time and they are indeed more expensive. And lastly, in terms of scientific innovations, we do need to consider the digital innovations, such as the contact tracing apps, which emerged in many countries quite independently, and this alerted people about potential exposure, but also AI and machine learning, which also played a key role in drug discovery, by helping to identify potential treatments and optimizing vaccine designs. And lastly, in terms of public health measures and compliance, it is worth pointing out that all of the scientific uh, breakthroughs that occurred would not have been useful if the public wasn't um, wasn't adopting the recommendations made by the government and by the relevant health institutes. And among the key public health measures, of course, we had masks, hand hygiene, and physical distancing, and the infamous lockdowns and quarantines to limit the movement during severe outbreaks. However, a key struggle in terms of preventing the spread of the disease is the larger population having a difficult time to either comply or to implement the recommended measures. For example, people living in crowded conditions, or people that would do essential work for the infrastructure. In both situations, isolation was difficult, and the spread of the virus was inevitable. Uh, just as important, and perhaps even more worrying, misinformation, whether on social media or just and between people in that would protest against the implemented measures, those could lead to resistance regarding adoptions of measures like masking or vaccines. And here comes the role of the public trust. People need ultimately to trust in science and to trust the government that they do take the right decisions. And unfortunately, that can be quite difficult. Sometimes the government can make mistakes or even contradict what the scientists recommend. And in scenarios like that, the population can have quite a difficult time to adopt these much needed measures. And in order to overcome these challenges, effective communication campaigns were absolutely critical for the good of everyone. Here I would like to invite you to an open discussion before we can look back at certain aspects of what we have talked about earlier. And I would really like you to contribute and engage and try to um, see how we perhaps can prepare for the future pandemics based on the information that I have presented you with so far. So to start with, let's just have an open discussion about which innovation or strategy had the most significant impact during the pandemic and why. Right. So answers are coming in the chat. Let's start with the very first ones. The lockdown was key in stopping the spread, but was widely unpopular. That is true. Um, People just didn't like being confided and being told what to do. And in various scenarios, that wasn't a feasible option for most of the people, either due to economic constraints or because of their living situations. Easy access of COVID tests, such as PCR, because this allowed us to identify and isolate infected people. Absolutely true. Quick development of mRNA-based vaccines, which can help in the future. Public health and innovation in vaccination. Here's another interesting question. The rapid development of vaccine as that directly kills the virus instead of just inhibiting the effect of it. Yes, I see that quite a lot of the answers revolved around mRNA vaccines. And here, what I would like to say is that the implications are beyond just this pandemic. Significant efforts have been made to use this technology to address other diseases, whether it is Parkinson's, Alzheimer's, or even other viral diseases. And the biggest step that happened during the pandemic was that for the very first time, mRNA vaccines were approved and used en masse. And this sort of approval by health organizations essentially encouraged a new age of vaccine development, so we can prevent many diseases and even help them eradicate. Right, moving on to the second question of the open discussion. What do you think should be prioritized in order to prepare for future pandemics? Yes, absolutely. Maintaining of public trust, that is key. More and more we see that we live in an age of fake news, of disinformation, and scientists have a much harder time to get their message across and to gain that trust from the wider population. And indeed, having this sort of relationship between the scientific community and the wider public is crucial. Scientists need to effectively communicate what they do, especially since their work is funded by taxpayers' money, and communicate their work in a way that simplifies complex scientific concepts and makes them readily understandable by people that are not necessarily trained in science. Another interesting answer: developing tests quickly so infected people can be isolated. Yes, absolutely. Just as the PCR tests and the lateral flow tests were developed in case of new viral diseases that present a high risk, we do need to have this means of identifying the disease in a reliable way. Another answer: I'm torn between the development of vaccines, maintaining public trust and compliance. As mentioned, without the compliance of the public, a disease cannot effectively be tackled. That's correct, and I would say that the answer is probably a combination of everything that we have talked about so far. Um, on the one hand, even if you have compliance, if you don't have the medicine, you cannot tackle the pandemic, or you can only postpone the moment when the virus enters the population. For example, we can take New Zealand. They were able to through lockdown, lockdown in the initial days, and then by closing the borders, to essentially have 0 cases of COVID-19 for more than a year. However, as time went on, the breakouts of COVID-19 epicenters did occur. And perhaps because the government wasn't necessarily well prepared to or well stocked up with vaccines, their response to that crisis might have taken a longer time. Whereas in countries like the United Kingdom or the United States, we did have the lockdowns, but we also invested a lot of money, of time, and of resources into developing these vaccines, such as the AstraZeneca vaccine deals. And when these were produced, um, um, let's say the United Kingdom or the initial countries that secured the funding, they had early priority in receiving the vaccines. But of course, when something like that happens, there are ethical problems that do arise. For example, let's imagine poorer countries that perhaps do not have that spare funding available. That means that they would end up being the last on the list receiving these vaccines. So thinking about all of these factors, such as ease of access and distribution worldwide of vaccines, of drugs, that is of utmost importance as well. More widespread education on the science behind things, like how the pathogen spreads, and therefore the public health measures that are put in place, and the potential treatment options to allow for more effective compliance. For example, some people wearing mesh masks for the aesthetic and to comply with the laws, when that doesn't really prevent droplets for spreading. Yes, that is a very good point. We have indeed seen in the early days of the pandemic people making fashion statements, sort of complying but not really to the rules, and thinking that just by doing certain things that would protect them from getting infected, such as the common belief, although wrong one, that just by taking vitamin C you can prevent getting a cold. Similarly, with the pandemic, maybe like people thinking, okay, I'm going to wear a scarf that is going to prevent me from getting COVID-19. Giving the public clear instructions and improving positive communications. This would be tough because people would probably be more likely to rebel against measures, such as lockdowns, quarantines, as this became less effective. There would likely be less trust because of high up MPs rule breaking in the COVID-19 pandemic. Yes, it is quite disheartening and disappointing when those that we look up to do not follow the recommendations that they would like to see being implemented in the wider society. One rule for the many, another rule for the few. And hopefully, as the society moves forward, people, especially the people in power, will have learned the lessons that trust is paramount in trying to keep at bay the spread of a pandemic. Right, and moving on to the last question: if you are leading a response team, what would be your focus? Try to think in terms of what kind of organization would you be part of? Would you be the responder for your school or maybe for the local hospital? Try to be imaginative here. Another answer came in. My focus would be mostly towards public health measures, as this is vital to curb the spread of the pandemic. Also, the impact of the healthcare workers, as they need to be able to deal with the surging cases. In addition, global collaborations which can boost development in vaccines and other innovations. Yes, precisely. Being able to engage with scientists from all over the globe that can facilitate the discovery of new treatments. And I can give you an example where researchers from my own group were part of this initiative called the Moonlight Initiative, where scientists from all over the globe developed a platform where they can screen a large number of drugs against a key drug target. And being able to do that systematic screening of many drugs, of course, that is a logistical challenge in its own right. So, in order to achieve the results that you want, you need to be able to coordinate the efforts of many different scientists. For example, here in Oxford, we were able to test the inhibitory effects of various small molecules against the main protease enzyme. And just going back to one of the slides, so I highlighted the main protease enzyme shown here, and that was part of our contribution to the development of a new drug. But then, of course, like on the other side of the globe, we did have collaborators where they would test the drugs that were either identified during the screens or developed by us during doctoral or postdoctoral research projects, take those drugs and test them in infected cells. The pipeline of development from designing it or screening it, usually to actually having it in the clinic, starts in the following manner. So first, thanks to the innovations that we had with AI and machine learning, we can virtually screen millions of different compounds against against certain enzymes, and we can virtually assess whether they would fit well in this active site. And if their predictive score indicates that they would indeed be effective at inhibiting the enzyme, the next step is to test the drugs in vitro, essentially against the isolated enzyme, and then see if we can reproduce that sort of inhibition. But then, just because a drug inhibits the enzyme doesn't mean that that drug would end up killing the virus. So what we need to do next is to give the drug to infected human cells, cells that were infected with the SARS-CoV-2 virus, and see if the virus dies off or if the virus doesn't replicate anymore. And if that does happen, then we can move on with the drug and do trials in mice or higher mammals. And when doing that, we of course need to consider various physical chemical aspects that would make a molecule a drug that would also work in humans. For example, the drug would need to be soluble. It would also need to be stable under physiological conditions, not to be degraded by liver enzymes. And of course, we don't want a drug that, let's say, if it does get metabolized, the metabolite, in other words, the byproduct that is being formed, ends up being toxic towards the host. And once we reach that stage, of course, we can take it to clinical trials. There are various phases. And then once it is approved, it can be delivered and distributed to the wider population. But another challenge to making this possible is, of course, the synthesis and the production of the drug itself. Sometimes it is rather easy and facile to produce a drug, let's say on milligram scale under laboratory conditions. However, if we want to have a drug that will potentially be given to millions of people, suddenly we need to think of producing this drug on kiloton scales. And that is a challenge of its own. But here I would like to invite you to another open discussions and ask you about the drug that I'm highlighting here, nirmatrelvir. What do you think you could modify about this drug to ensure that in the future we have new modes of action, or we can have similar drugs that end up being potent against mutants of the SARS-CoV-2 virus? Just feel free to drop in the chat any ideas. All right, well, I can start helping you here. If we look at the molecule, we can see distinct parts. And these distinct parts would be defined by amino acid residues. An amino acid or like apologies, fragments that end up forming amide bonds. And that is what would give rise to a peptidomimetic drug. And in our instance, I was mentioning earlier, this group here, the nitrile group, is our warhead. And it is our warhead because it can irreversibly bind to a key residue at the active site, which is a cysteine residue. And the cysteine residue attacks the nitrile to form a covalent bond. Now, in terms of developing new drugs, what scientists do is trying to identify other alternative groups that perhaps have similar physical-chemical properties, but end up inhibiting the main protease to the same extent. And instead of having a nitrile group, we could also have an alkyne group. It is a biosteric group in terms of it occupies the same space. It has somewhat similar properties, but it can also bind to our cysteine residue. And when it binds, that ends up being an irreversible covalent bond. In terms of drug design, scientists have also used alpha ketoamides, such as the one that I'm drawing here. or aldehydes. So essentially what you want as a reactive warhead is a group that on its own is somewhat unstable, high energy, and that can react with other molecules. But you don't want it to be too reactive because in that scenario, you could have undesired side effects. You want your drug to be effective against the SARS-CoV-2 virus, but you don't want it to be effective against human enzymes because in a scenario like that, you can end up with cytotoxic side effects, and the host could end up having severe side effect health related issues. Um, I hope that you have found this session interesting, inspiring. If you have any questions in the chat, I'll be happy to answer.